Hi everyone. Today we are going to be talking about recombinant DNA. First we ask the question, what is recombinant DNA? Recombinant DNA is the method of the joining of two or more molecules to create a hybrid. The technology is made possible by the use of two enzymes. The first kind is the restriction endonucleases, also known as the restriction enzyme. And the second one is the ligase enzyme, which we all know. So the restriction endonucleases, also known as the restriction enzyme, recognizes a specific sequence in the DNA and cuts it or close to that sequence. And then the ligase attaches the plasmid or we'll talk about it inside the PowerPoint. We'll get to it. Here shows the recombinant DNA technology and what the recombinant DNA is made of. So here it shows that the restriction enzyme cuts the specific sequence or near to it, and then it forms a recombinant DNA after the process. And it shows here that the recombinant DNA is made out of a vector and a DNA fragment. So there are 5 main steps to the making of the recombinant DNA. There are 5, and I will list them before we explain them. Isolating the genetic materials, restricting enzyme digestion, using PCR for amplification or also polymerase chain reaction, ligation of DNA molecules, and finally inserting the recombinant DNA into its host. So first, let's talk about DNA isolation. There isn't much to say about it, but it's technically that the DNA is isolated in its pure form, which basically means that it's divided from the other macromolecules. The second step is the cutting of DNA. So for this step, the specific enzyme, which is the restriction enzyme, plays a very vital role because it helps to identify the location in a designated gene, which is then introduced to the vector genome. The third step is the amplifying of the DNA. So in this step, copies of genes are amplified through either PCR or polymerase chain reaction. And it essentially is the process to increase a single DNA copy into several DNA copies after the desired gene of interest is cut using the enzyme of restriction. The fourth step is the joining of DNA. So here, the vector and a section of DNA are joined together with the help of a specific enzyme, which we all know as, drumroll please, the ligase enzyme. Here is the plasmid and the recombinant plasmid that we said we will get to in the PowerPoint. So there is a plasmid and there's a target gene, which is like an insulin gene or the protein gene, etc., etc. The plasmid is digested with the restriction enzyme, and the target gene is attached to it. And then it's joined together by the ligase, and then it forms a recombinant plasmid. Now we will talk about the recombinant DNA uses. So recombinant DNA has been known to be very effective, its technology in particular, because it is known to produce vaccines and other various human proteins in a specific area, which is microorganisms. So these are such as insulin and growth hormone, which are used in treating diseases, including cancer. It is also used to produce some clotting factors for treating, quote unquote, specifically hemophilia. I don't know how to pronounce it, but it's hemophilia, and in the development of the gene therapy. Here is an example. So, for example, insulin is regularly produced by means of recombinant DNA within bacteria. A human insulin gene is introduced into a plasmid, which is also known as the targeted gene, which is then introduced into a bacterial cell, and then the bacterial cell takes it and uses its cellular machinery to produce the protein insulin, which then can be collected from the body using either blood transfusion or anything, and then distributed to patients that don't have it or have problems or have diseases. And that's it. I hope you enjoyed.